Okay, hey everyone. So this is the sodium and osmolarity homeostasis lecture. Um, this one is going to be a little bit tougher and I feel really bad. So I'm actually in a bit of a rush to get this video out. So if it's really bad, and you guys are watching this before Monday, send me an email and say, hey, I'm still pretty lost. And I will try and put out a supplemental video going over all the hormones. I'm just going camping. I need to leave in like 20 minutes. So this video is going to be really short, you guys. I'm sorry, but hopefully it'll at least get you an introduction. So when Dr. Busey talks about it on Monday, it'll be more in your head. So everything we've talked up to this point is like establishing what the kidney is, basic physiology of the kidney. This is the payoff step. So this lecture does contain a lot of material. I would highly recommend going over multiple times. So you're going to learn about different hormones. You're going to learn what countercurrent is. And I'm not going to explain them super in depth because I already said I got to run. So I'm so sorry about that. Please read in your books to supplement the information here. It has it all in there. So please, please, please read. Please email me. I got to go. So here we go. So understand water and sodium homeostasis. That's kind of generic, but we'll show you how that's going to happen. Understand the origin, target, and mechanism of action of the relevant hormones. So that's going to be ADH, and this should be a parentheses. Oh man, you guys can see how much of a rush I'm in. Okay, and then now that you know what the current does, understand countercurrent. And so I'm going to just say this now because when I took this class, countercurrent right here just confused the heck out of me. So all we're talking about, countercurrent kind of gives us the reason why we can get so much of these different steps. So we haven't told you like the mechanism by which they're happening. Like sure, in reabsorption, we told you like, okay, well, sodium goes here and then it gets pumped out here, and maybe water follows sodium, and then urea would follow because of concentration and everything gets filtered. So you know what's happening, but this countercurrent kind of gives you the why of reabsorption and secretion are occurring. So not excretion or filtration, I'm sorry, but countercurrent would be reabsorption and secretion. Don't confuse yourselves. You already know what this is. I promise, promise, promise. You know what reabsorption is. You know how secretion works. We're just filling in a couple physiological blanks that we haven't taught you yet, okay? First, I wouldn't memorize any of these numbers. They're not important. But what we're saying here is that you do get water through your food and drink. You get it through metabolism, blah, blah, blah. And you have to maintain this, right? So if you're just taking in a bunch of water, you're going to explode if you're not getting rid of it. So your body has to like take in the same amount that it's getting rid of. We don't really store water all that much, right? So whatever's flowing in our plasma will get filtered. It's also good, like get rid of anything that's in that water that's floating, or any solutes in that water, okay? So the main way we do this would be through the urine, but also through feces, as we'll talk about in GI. Like you guys know this already. You get diarrhea sometimes. What causes diarrhea? Well, that's excess water. So your body is getting rid of the water in the feces and urine and insensible loss. To me, this just means like sweat or like even in respiration, maybe a little bit of water is coming out. But the main one we need to know is urine, obviously. So this is fluid and electrolyte balance. These charts always, to me, seem so hard when I first look at them. So I want you to like blank out this right side of the screen for a second. So first we're just doing a decrease in blood volume leads to a decrease in blood pressure, right? How do you get a decrease in blood volume? Well, you could urinate, you could get stabbed and lose a lot of blood. You could, any of those things. Like if you have a bruise and you're bleeding internally, you're losing blood volume, lowers blood pressure. Okay, that volume, then there's receptors in the atria and carotid and aortic baroreceptors. Don't get confused here. So we already talked about some of these volume receptors, right? So we talked about the myogenic response 2 lectures ago. If you don't remember that, I'd look that back up. But the myogenic response is when um, the afferent arteriole can sense how much is coming through and it can constrict or dilate a little bit to regulate the GFR. So the kidneys, while this box is saying, hey, the kidney is not the only thing that can do that. The atria does it, carotid and the baroreceptors, okay? Then they trigger homeostatic reflexes. This term is super generic to me. I don't love it, but what it's trying to show you is like, okay, this decrease right here, you're going to start releasing hormones. That could be ADH, that could be aldosterone, maybe even epinephrine. And so you're going to get that kind of release, which leads to everything down here. So you can pause the video and go through. I really think these steps make sense. Like your behavior is going to change. Okay, behavior, now you want more water, that increases, you get an increase of blood pressure, hormones talk to the kidneys, hormones talk to the cardiovascular system, and kind of the exact opposite over here. If your blood volume gets too large, talk to the cardiovascular system, say, please stop pumping so much, we can't handle this. Kidneys, okay, get rid of this water, push some salt out so osmosis can work in our favor. So that's all this slide is showing. Review question, please take time and go over this. The right answer is, okay, you can pause the video and I'll give you the right answer in just a sec. Okay, C right there, inulin, right, doesn't get secreted or reabsorbed, just it's filtered and excreted. Okay, GFR, this is like a tube, so this is the GFR. Whatever comes through here is representative of GFR or glomerular filtration rate, okay? So this was kind of confusing the first time I looked at it, but I do think it is gonna be helpful. So we can decrease the GFR here, and that would then decrease the bodily fluid if we lose more in the urine. Or maybe we shut this right here and we don't lose any in the urine. So GFR just gives us a little way to regulate the balance here, right? Can't do it all. It is regulated by hormones, regulated by reflexes like the myogenic response or the tuboglomerular tuboglomerular response. I just cannot say that, you guys. Okay, aquaporins. So this is the first big, huge thing I would know from this lecture, and I would never forget it ever again. So aquaporins are little channels that allow water to get through, okay? So we insert aquaporins in the basolateral membrane when we want to maintain more water, okay? It allows water to get through these cells and shoot out the backside, okay? So what you can see here is vasopressin, that's the hormone to know, and I hopefully think that you guys will know that vasopressin is ADH released from the posterior pituitary, but it is synthesized in the hypothalamus. It's a good review. It comes down here and it acts specifically on these kidney cells, okay? So if you get that, you're in real good shape. So all you really need to do here is say, okay, well, vasopressin comes, binds right here, makes cyclic AMP. This is a good thing to know. I don't know if you're specifically tested on it, but the mechanism by which it does it, great. Okay, you get the signal from whatever cyclic AMP does. That signal actually ends up inserting these aquaporin channels into the membrane, allowing more water to come through. Okay, so perfect, I would know that. Make sure not to forget aquaporins are associated with vasopressin because we have to learn about aldosterone, which does pretty much has the same results, but takes a very different pathway. So one more time, aquaporins, vasopressin, channel that allows water to come back in. So directly works with water, okay? Ooh, I didn't know there were, sorry guys, here we go. Oh, and this I would also know, sorry, I forgot to say this. It's in the distal tubule and the collecting duct, so that's more towards the end. And then aldosterone, if I'm not wrong, is only in the collecting duct, but we might need to double check me on that later in this lecture. But just know this is like the last stitch after you've made it through the whole nephron, then you're getting the action of vasopressin. Okay, this is kind of confusing. So I mentioned it in the lecture video from Friday, if you guys have watched that, in the very first kidney lecture, but there are different portions of the nephron. So this would be proximal, loop of Henle, so descending loop, ascending loop, excuse me, and the collecting duct. As you can see, the descending loop, I would write this down, not necessarily for this class, but for MCAT or other tests, is very permeable to water. So water is coming out, okay, in the descending loop. I think in the next lecture, you learn a little bit more about specific cell types that allow this to happen. So hopefully you guys have realized that the kidney lectures, they are intense. Kidney physiology is real tough. So it's broken down into 4 lectures and just each one adds upon the next one. So if you guys got lost, please go back and watch the first one about nephron anatomy. So descending loop, you get water that can come out. The ascending loop is impermeable to water. Highlight, asterisk, whatever you got to do to remember that. Water's not moving in this ascending loop. Can come out over here, stuck in here. But what does move is solutes like potassium, chloride, sodium. And why is that important? Well, we remember osmosis, right? So the water can't move here. We do not allow it to get through. But if we change the osmolarity of the water coming back up here, all of a sudden the water will start flowing out in the collecting duct in the distal tubule when it is permeable. And it could be permeable under ADH, which will insert aquaporins, or by aldosterone's mechanism, and we'll look a little bit more into that. So I would know that right here, it's kind of cool. So The kidney is really the only place in our body where we can safely have an osmolarity of up to 1200 milliosmoles. So normal blood is 300. So when we go down into the medulla, we can get up to 200 milliosmoles. That would be if you're super ridiculously hydrated and your urine is completely brown at this point. Um, and 300 would be 300 or less. You can actually go from 100 milliosmoles, super dilute. That's you're very, very hydrated, to all the way down to 1200. And that's through this really cool mechanism of the kidney being able to loop down, be permeable at one point, not permeable at the other point, and then allowing the water to move here really allows us to get that different osmolarity changes. Super cool. I would watch other videos if you want to on this. It's kind of hard to understand, but just know as you move down into the medulla, you can change the osmolarity. You can make it more, um, have a higher osmolarity, and then it comes back up into the cortex and it lowers the osmolarity. Okay. So down lower in the medulla, that means you have a most likely a lower or higher amount of solutes. Not necessarily, but you have the potential to do that. Okay. ADH puts final touch on urine specificity gravity. So we just talked about that, right? This is introducing the thought of countercurrent, which we talked about in a few slides. But what's happening here is you have to remember we do so many pictures of just something like this, but the nephron isn't alone. There are capillaries all over every single part right here, okay? And that's kind of what's being shown here. So all of a sudden, you're up here and you're at like a super, super low osmolarity in the tubule, okay? But the vasa recta, the capillary that surrounds the nephron, actually has gone through a crazy amount of concentration. So it's all the way at 1200 milliosmoles down in the medulla. So what's happening is the flow of blood is like this, up like following the arrows. The flow of urine is out like this. So what's happening is you're getting more and more water coming from here into the blood. You're saving the water, and then the blood takes it off, okay? So that is how we maintain osmolarity. Hopefully that makes sense. So I'll do it one more time. So as you decrease and you're getting closer and closer and closer to actually being excreted out of the kidney through the urine, your osmolarity can go up. It doesn't necessarily have to. If you're super, super hydrated, none of this might occur, but the kidneys can do it. Every one of our kidneys, unless we have severe kidney damage, can regulate our solutes like this, and it's super important, okay? So water will come out through aquaporin channels that ADH has set in the collecting duct, and it will go over into the vasa recta. That's what's pulling it over, right? So it's at like a, um, the water is at a certain concentration here versus here. It's going to go across into the blood, okay? So that's super important to realize. And you can also, in the absence of vasopressin, you can block it so water can't come out. So that would probably be when you have a normal solute osmolarity, you don't get ADH signaling. And so then your water comes out of your urine and you come out seeing just really clear urine that is shows you have a lot of water in your system. Okay, I'm not going to go over this. Please go over it though, just to make sure you understand everything. Shoot me an email. Sorry, I'm just running out of time here, you guys. I don't think this is the most important thing in this slide. Countercurrent. So this happens so this is a limb. Forget kidneys for the next 30 seconds. Countercurrent occurs everywhere in our body. So look down at your hand, and you know from anatomy, for those of you who have taken it, that the arteries and the veins usually flow pretty close to each other, and they kind of turn around. Like right here, this would be like the capillary, right? And then the vein would be more cold blood. So just what a countercurrent is it can be heat exchange, but we also just saw it can be solute exchange, okay? So this is the canonical example that happens everywhere in our body. Heat exchange at the end of our limbs, end of our fingers, end of our toes. We get this countercurrent if they're further apart. Excuse me. Then you lose heat. If they're close together, then you kind of keep the heat. Now we get to talk about countercurrents in the kidney, which are just a little bit more complicated. But if you understand what we talked about 3 slides ago here, and then understand countercurrent, then you're good to go, you guys. So just remember, like we said at the beginning of this video, this is why we are allowed to have reabsorption in different levels of that throughout the kidney, because the blood is offsetting whatever's in the kidney. So you start here, right? Let's say this is, the descending loop of Henle. Water is coming out, okay? And it's going into the vasa recta, which is returning to systemic flow. And the vasa recta during this point, it started over here. So it's going the opposite direction. Hopefully you can see that by the arrows. This is going this way and the blood is going this way. So new urine that's entering this system is pairing with old blood. Not like, I hope that makes sense. This is relative terms to like what was filtered through the glomerulus. So stuff that is just recently filtered is pairing with really old blood to make sure that the blood is staying like in homeostatic means, staying with a good osmolarity around 300 milliosmoles. Because if the blood doesn't stay like that, once it gets out of the kidneys, you have some serious problems, right? That would throw off all your Nernst potentials, all that nonsense. You could have issues if you cannot maintain electrolyte balances. And you've heard of this with like patients who have kidney failure or organ failure. Sometimes balancing their electrolytes when the kidneys aren't working is the hardest thing you have to do to keep them alive. So just realize this is super important. So you're getting countercurrent. So water's coming out of the descending loop. Water can't move, but sodium can into this one. So what we're saying here is in the ascending loops, all these electrolytes are moving into the blood, thus making it very high in osmoles. Have a very high osmolarity, my bad. And now all of a sudden water can come back in. So if you're looking just from the perspective of a blood cell, it sees all these electrolytes following it, all these electrolytes. It's like, oh man, this environment's really inhospitable. Then it passes and it's now working along with the kidney. And now it's like, oh, water, water, water. Okay, now we're good to go. So that's how I like to think of it. And then just think opposite flows. That's countercurrent. That's probably the hardest part of this lecture. At least it was for me. Dr. B will for sure cover this because it is something really hard to understand. Not going to go over this. Last thing we need to do is aldosterone. So aldosterone is a steroid hormone. I had to remember that. It's a steroid hormone. Where do steroid hormones bind? Well, they actually bind inside. Steroid hormones just blast their way through the lipid membrane because they are lipophilic, whereas ADH was, I think it's a neuropeptide, meaning that it has to bind here on the surface on its receptor. Aldosterone gets in, activates transcription, and now what you're doing is you're actually allowing for electrolyte channels to come in. Now, why is that helpful? Well, electrolyte channels help with the flow of sodium and potassium, as we see here. Sodium back into the blood, potassium out. And what happens is water loves to follow its sodium, right? So that's how you can think of this being important for water flow is if the sodium's reabsorbed, the water's being reabsorbed, and that allows for you to keep more water. Let's see, is there anything I missed here, you guys? This is in the distal nephron. So I'd say distal tubule and collecting duct as well. I think we're all good there. This is the last thing about aldosterone. We have to learn the RAS system, the renin-angiotensin system. If it's not covered specifically in the next lecture and I was supposed to cover it here, I will for sure throw it in because I do know you will be tested on that. So please don't worry. We'll talk about P cells next time and all that. So that's going to be it for my presentation today. I'm so sorry that was so rushed. Play it multiple times, send me emails. I can do another video if you guys are so confused. Just let me know. I'll be back on Sunday. So just go ahead and send me your complaints, I guess. Okay, have a good day.